Good morning, students. We will be continuing with Science 2, chapter number 8, Biotechnology and Cell Biology. So, today we will be starting with the third application of biotechnology in human life, that is, human health. Now, we know that diagnosis and treatment of the disease are the two important aspects of the human health management. Diagnosis means to identify what disease and treatment of the disease. Now, biotechnology helps us to identify the role of the gene in disease of a person. For example, diagnosis of diabetes and heart disease have become possible even before the onset of the symptoms with the help of biotechnology. Onset means starting of the symptoms. Diagnosis of disease like AIDS, dengue can be done within few minutes. Hence, treatment can be done at the earliest. Various medicines are used for the treatment of disease. For example, the hormone insulin is used in the treatment of diabetes. In the olden days, insulin was collected from the pancreas of horse. But nowadays, due to advance in biotechnology, insulin is prepared with the help of bacteria. We saw that in the previous chapter when we were learning about microbiology. For this purpose, human insulin gene has been inserted into the genome of the bacteria. Various vaccines and antibiotics are produced in that way. What is the meaning of the word genome? Genome basically means the complete set of genes or genetic material that is present in the cell of an organism. Biotechnology in human health. How is it useful? It produces better vaccines, enzyme replacement treatment for genetic disease, better and customized medicine, faster and more accurate diagnostic, gene therapy, forensics, and regenerative medicines. We will be seeing some of it. Role of biotechnology in vaccines and vaccination. Vaccine is the antigen containing material given to acquire either permanent or temporary immunity against a specific pathogen or disease. This is the definition of vaccine. We have learnt about it in the lower class. What is the meaning of the word antigen? An antigen is a substance that causes our immune system to produce antibodies against it. This means our immune system does not recognize the substance and tries to fight it off. That is the purpose or process in which a vaccine works. We know in the olden days, vaccines were prepared with the help of pathogens, that is, microorganisms causing disease. A completely or partially killed pathogen were used as vaccine. That means it was injected in the body and the body produced antibodies to fight it, either mostly before the onset of a disease or the moment to protect a person from a disease. But sometimes there were chances of getting the disease in case of some person. Hence, as an alternative, scientists tried to artificially produce vaccine with the help of biotechnology. For this purpose, scientists produced the antigen in the lab with the help of gene isolated from the pathogen and used as a vaccine. Thus, safer vaccines were produced. Now, proteins which act as antigens are injected in the pure form instead of injecting the killed or the semi-killed pathogen. Because we are not using the pathogen anymore, we are using a vaccine which is created in the lab. These proteins keep the person away from the disease by keeping the immune system active. Thus, injecting the antigen is the safest way in vaccination. Vaccination produced with the help of biotechnology are more thermostable. That means they can withstand temperatures and remain active for a longer duration. For example, vaccines of polio, hepatitis, etc. Edible vaccines. That means vaccines that can be eaten. Work on production of edible vaccine is in progress, and presently potatoes are being produced with the help of biotechnology. These potatoes are called as transgenic potatoes. These potatoes will act against bacteria like Vibrio cholerae and E. coli. Consumption of these raw potatoes generate the immunity against cholera and the disease caused due to E. coli. So what happens in this case? In this case, if you see the diagram, that they inject virus in the plant, and then it creates a transgenic plant. Then the virus is infects the pieces of potato leaves. Then the entire plant is developed from the leaf pieces containing genes of human pathogen, and consumption of the raw potatoes will help to develop immunity against that particular pathogen. Another image showing an edible vaccine, a edible vaccine which is put in a carrot. See the do you know paragraph. Here it's written Indian Institute of Science has developed a transgenic variety of tobacco. That means it's a edible vaccine. If the cattle feeds upon the leaves of this plant, they will not get a viral disease called as rinderpest. So there is a lot of research going on related to vaccine and edible vaccine. The next thing is how biotechnology is useful for the treatment by production of hormones like insulin, blood clotting factors, etc. Then we have something called as interferon. This is this group of small protein molecules is used in the treatment of certain viral diseases. These are produced in blood. However, nowadays with the help of biotechnology, transgenic E. coli are used for production of interferon. Then the next thing in human health is gene therapy. Gene therapy is used to treat genetic disorder in somatic cells. It has become possible due to the help of biotechnology. For example, PKT arises due to genetic changes in the liver cells. It has become possible to treat it with gene therapy. This method is called as somatic cell gene therapy. All the cells except the sperm and the ova in the body are called as somatic cell. So what is gene therapy? Gene therapy is a therapy in which the genetic manipulation is taken place and the it is treatment takes place. Now this for this particular we use the somatic cells. Now somatic cells are all the cells of the body except the reproductive cells. They are called as somatic cells. This is a pictorial representation of gene therapy. Gene therapy is an experimental technique that uses genes to treat or prevent disease. In the future, this technique may allow doctors to treat a disorder by inserting a gene into the patient's cell instead of using drugs or surgery. Cloning. Reproduction of replica of any cell or organ or an entire organism is called as cloning. Replica means to create something which is exactly similar. Cloning is of three types. One is called as gene cloning, one is called as reproductive cloning, and the third one is therapeutic cloning. Here we are going to learn only of two types. We are not learning about gene cloning. Gene cloning basically means to replicate the gene or the DNA. What is reproductive cloning? Reproductive cloning means a clone can be produced by the fusion of a nucleus of somatic cell with the denucleated or enucleated ovum of anybody. Thus, there is no need of the sperm to produce the new organism. So, what we do in this case, you take the somatic cell and from that, somatic means non-reproductive cell, and remove the nucleus. Then you take the egg cell and from that you remove the nucleus. Then you combine the nucleus of the somatic cell and you combine the inside the nucleus and then you produce the clone. This can be then implanted in the surrogate mother. So if the embryo is implanted in the uterus of a surrogate mother, a new cloned organism will be developed. This is called as reproductive cloning. क्या बोल This is an example of reproductive cloning. A sheep named Dolly was born in Scotland by cloning technique on 5th July 1996. Nucleus from the udder cell of the sheep of Finn-Dorset variety had been introduced into the enucleated ovum of the Scottish sheep. Then the ovum was allowed to develop into the uterus of the Scottish sheep and thereby the Dolly had been born. It was showing all the characteristics as per the chromosomes of the nucleus, and any characters of the Scottish sheep was not visible because it was the exact technique of cloning where the ovum, since the nucleus was removed, there was no characteristics of the ovum of the mother. The diagram shows how it was done. Therapeutic cloning. Now, this is basically for the treatment of diseases. Stem cells can be derived from the cell formed in the lab by the union of somatic nucleus with the enucleated egg cell. Various diseases can be treated with the help of this stem cell. What we have to understand in this cloning process is what we are doing is from one cell you are removing the nucleus. So there is no characteristics of that particular cell. Whereas in the other cell you are just taking the nucleus. So you are exactly replicating those characteristics. So you can create stem cells, and we saw the stem cells can be used to develop different cells which can be used for treatment. So similar to cells, genes can also be cloned and millions of copies of the same can be done. This is called as used for gene therapy, or this is called as gene cloning. Controlling of inheritance of hereditary disease, continuation of generation, enhancing the specific tendency may become possible due to cloning technique. But there is a worldwide opposition to human cloning on various issues. Because what happens is human cloning till now is not being allowed. But by cloning method, we are able to control a lot of things. For example, suppose you know that in a particular family, everybody is having diabetes or having high blood pressure. Then right before the onset of the disease, we can stop that. So they have given you certain examples like insulin can be treated for diabetes, then interferon can be for viral infection, then interleukin for cancer. So there are various hormones or which can be utilized for treatment of the diseases. This is a comparative chart which is showing normal development as well as reproductive cloning and therapeutic cloning. During normal, what happens? The male gamete and the female gamete unite to form a zygote. So the zygote gets the characteristics of both, and then from there you are getting an adult. Now here the adult has characteristics of both the male and the female. In case of reproductive cloning, what is happening? You are taking a nucleus of one adult and you are taking a cell without the nucleus, a somatic cell. And when you combine, so the cell that is formed, or the zygote that is formed, is only showing the characteristics of one parent. Then that is called as reproductive cloning. In therapeutic, what happens? The process is similar, but it is not used to create an organism. It is used to create stem cells or embryonic stem cells. And these stem cells can be used to form different cells, maybe a liver cell or a heart cell or something like that. And then that can be useful for treatment. Thank you. We'll be continuing in the next lesson.